Hello, everybody. Thanks for joining us today, and thanks to LabRoots for inviting us to talk about the next big question, genomic surveillance of SARS CoV-2. My name is Gary Schroth. I'm a Vice President and Distinguished Scientist at Illumina. I've been at Illumina for 16 years now, working on applications for our next generation sequencing sequencers, especially working on RNA applications, which is how I got into viral detection and viral sequencing. And so what I want to talk about today are variants and how important that has become to this phase of the pandemic and how next generation sequencing is helping us get through this. So NGS, or next generation sequencing, really does have a unique role in our response to COVID-19. It's been used to monitor the spread. As you can see by those worldwide patterns there, you can see variants kind of moving around the world. But probably the second one here is the most important, and probably what we're going to talk about the most today, is identifying strains that have increased transmissibility. That has become really, really important and a topic in the news for the last few months. The variants, as they're just referred to on every newscast, are determined by next generation sequencing. It's also important to understand the way the variants might affect PCR diagnostics. I'm not going to talk about that too much, but essentially, the companies that create PCR diagnostics need to be very cognizant of which mutations are out there in the population, as that may affect the behavior of their amplicons used in PCR. And then another area that's not super important right now, but you can imagine how this is going to be more and more important over the coming year or maybe even years, is to identify the risk of vaccine escape mutants. Sooner or later, some of these vaccines are not going to work quite as well on certain strains, and certain strains will prosper in the presence in the background against certain vaccines. And so that's going to be a really, really important piece of information that we will need from a public health point of view over the next few years. So genomic surveillance, as it's called, this sort of strain surveillance, is really important to monitor, as I said, new strains and monitor trends after interventions such as vaccinations. It's really interesting to think about, we know, especially in the UK and the US, we have a lot of sequence information now. We also have a lot of vaccination going on. And the question is, what will be the strains of this virus that are going to be around in a few months? Because it's undoubtedly not going to go completely away. Hopefully we'll keep the infection rates down low. But which strains will be kind of going through the population after vaccinations have happened? That's going to be a really interesting question. And at the state and local level, this information can be used to identify clusters, outbreaks, and understanding kind of the local epidemiology of the virus. Of particular concern are variants that are associated with the S protein, or the spike protein. This is the protein, as you can see there on the diagram. The receptor binding domain, or RBD, of the spike protein is the part of the virus that interacts with the human cell through the ACE2 receptor. And so that's like the critical point of contact with humans, and that for this entire virus. And so mutations in and around the spike protein and the RBD are extremely important and monitored very carefully. And so that's sort of why the UK variant that has become known, that was discovered last fall and monitored throughout the late 2020 in the UK, and you could see that it quickly spread across the entire country, is so interesting. And I'll talk about some of the specific mutations that it has. But a lot of these sort of variants of concern that are popping up now around the world are variants that have significant changes to the spike protein. So the B117, as it's called, the UK variant, was really kind of a little bit shocking when it was first discovered, because you could see in that diagram there in the middle, what we're showing is kind of the rate of evolution, the accumulation of variants over time for the first several months of the pandemic and the sequencing that was done. And then the variants around the B117 clade is shown there in that top part. It had a really unusually high number of mutations. So relative to background, where we were seeing 1 or 2 mutations accumulate over time, all of a sudden this variant appeared that had 16 or 17 mutations of which there's 7 or 8 that are right in the spike protein. So this was very worrisome, and also this variant seemed to be spreading quite quickly, right? So we now know in detail where all those variants are due to the sequencing of all the B117 strains around the world. But we also have used sequencing to discover other concerning strains like B1351, which is usually referred to as the South African strain. So this table, we try to summarize the 5 variants of concern that the U.S. CDC has on their list as of today. The 3 that I've kind of already mentioned, the UK variant, B117, the South African variant, B1351, and then the Brazilian variant, which is also very concerning, is called P1. And also, interestingly, just in the last few weeks even, 2 US variants from California have been added to this list of variants of concern. They are B1427 and B1429. What is really, really interesting about this is that these variants accumulated, in different places, in different people. We think, let me go back one slide. One of the ways that scientists imagine that you get variants like this, where you accumulate a lot of variants in one strain, is likely with the evolution within a single patient who had a long infection. And most likely this person was probably immunocompromised so that they had an infection for 20 weeks or longer. That allows the evolution of the virus to take place in all kinds of different ways within one host. And that's how you can probably accumulate a large number of mutations like this in a single genome. Anyway, back to the story. All 3 of these variants have those same kinds of patterns of large numbers of mutations. And in fact, which is really interesting and kind of a testament to the concept of convergent evolution, is that all of these 5 variants of concern share the D614G mutation, and a few others are shared by 3 or 4 of the variants of concern. So even though this virus has been out in the world in millions of people, millions of hosts, with all kinds of opportunities to evolve in unlimited numbers of ways, essentially at the end of the day, the variants that are causing us most concern do, in some ways at least, look very similar to one another. So how do we know all this information? A lot of research has been done on the virus using various methods in next generation sequencing. These are an overview of some of the workflows that we have worked on at Illumina. This is an RNA virus. So you can essentially do metatranscriptomics or shotgun RNA-seq on a nasal swab, and you can see the virus, and that's this workflow at the top here. Or you can use enrichment. Think of like exome sequencing or probe-enhanced enrichment of the virus. That works very well, and we have a series of products that can be used to study that virus, and in addition, other viruses. That's the sort of middle workflow there. And then we break it down by the library prep kit, the type of sequencer that is used, and then the type of analysis tool that is used for each of these workflows. But the method I'm going to talk about the most today is the Amplicon method. This is a method that we developed into an EUA, an FDA-approved EUA test. We call it the COVID-seq test. And more recently, we have made an RUO version of this protocol to be used for surveillance. And I'm going to talk about some of the details of that. We've also created lots of different Dragon pipelines. Dragon is our informatics pipeline methods, and that I'll get into more details here in a second. But we have several now interesting Dragon pipelines for analyzing COVID. So as I mentioned, the COVID-seq test originally, it's interesting. We started working on the COVID-seq test, now that I think about it, about a year ago in R&D in Illumina. We sort of dropped everything and started working on this COVID-seq test. And within 2 months, we had an FDA-approved EUA for this test and products on the shelf and everything to give to customers to study the virus. The test itself and the EUA approval is actually for the diagnostic answer of yes or no. Do you have the virus or not? the sequencing of the virus is sort of an additional benefit of using this. Since then, there are groups that use this for detection only, but since then, many more groups are interested in studying variants and studying the genome sequence. And so we have tweaked the protocols, as I'll talk about a little bit, in an RUO version of this that will give even better genomes than the original COVID-seq test, which was more tweaked for detection and sensitivity and speed. So the assay itself, I'll get into some of the details. This is the assay. The outline of the assay is very similar for the EUA and whether you're running it in the RUO mode or the EUA mode. It is based upon 98 amplicons. It controls that tile across the virus. It also has 11 human RNA controls, and it's designed to run anywhere from 384 all the way up to 3,072 samples in a single run. The biochemistry process of the workflow is, like I said, very similar between both the EUA and the RUO version, but essentially it starts with a sample that is RNA extracted, converted to cDNA, and then amplified using the COVID-seq primers. And down there at the bottom, you can see we use two pools of primers. These are based upon the Arctic designs that are out there in the community. The pool 1 and pool 2 are separated so that they don't interact with each other and that you can get overlapping amplicons during the PCR process. So the green pool 2 primers and the red pool 1 primers are in separate tubes. You amplify up the virus, amplify up the cDNA, then you pool them back together. And then what we do in our process is we then essentially shotgun sequence each one of those amplicons using a transposome method to fragment them randomly into a kind of a shotgun pool of sequences across every amplicon. And then we sequence it at very high depth on a NovaSeq, and we use that data to ascertain whether the virus is detected or not and to get the sequence. This process can be scaled up quite high on a NovaSeq, and this is the way the math works out. We have 4 sets of index plates, so that's 4 times 96. That gives you 384 different indexes that can be used on the system. Each set of 384 can be loaded into one lane, so one pool per lane. There are 4 lanes on each flow cell, and then the system can run 2 flow cells at a time. So that's 96 times 4, 384 times 4, and then 1536 times 2 gets you up to 3,072 samples per run. And the original COVID-seq test, we were going for speed, and so we were doing a very fast 1 by 36 base pair run. That run can be completed and analyzed in about 12 hours. For surveillance, as we'll talk about, we have extended the read lengths to 2 by 100 or 2 by 150. This gives you more genome coverage and actually more complete genomes, as we'll talk about. So this is some of the performance from the COVID-seq test, and this shows you how sensitive the test is, and how in the original version of the test that uses 36 base pair single reads, we still were able to get genomes out of quite a lot of the samples. So essentially, this test has a cutoff here, that lower red line, that if we detect at least 5 of the 98 amplicons, and we detect them with more than 10 reads per amplicon, we call that a presence, and that's the presence of the virus. If we detect more than 90 of the amplicons, then we reported out a consensus sequence in the original test. And that region in between, or 5 to 90 amplicons detected, was that was deemed positive for COVID-seq, positive for the virus, but not a consensus sequence. And so you can see that sort of starts to fall off between CT values of 30 and 35. Between 30 and 35, we still get at least some of the genome out, and that's well above our detection limit. What we have learned since the original test is that if you're going to use this biochemistry and this assay for surveillance, we have a few tweaks to the assay that help give it better genome coverage. The first one is that it's now known that if you do the same PCR process but with a 63-degree annealing temperature compared to a 65-degree annealing temperature in the PCR step, that gives more uniform coverage and better coverage. And so you can see that that's what is shown here. This is an example of 16 samples that were compared in terms of the percentage of non-N bases. So essentially, what percentage of the genome was covered by a sequencing result. And you can see that taking those same 16 samples and doing the process at 63 degrees, we got much better coverage, essentially 100% coverage of the virus. Whereas at 65 degrees C, we actually went down as low as 97%. It's still really excellent coverage, but there's still 3% of the virus is not covered. And for all of us who are doing this and go through the effort of sequencing a virus, ideally you'd like to get as close to 100% as you could. So in order to help us get real close to 100%, we also, in the RUO mode, are encouraging people to run the sequencers a little bit longer. So the 1x36 data is shown on the left, and this is just a progression of genome coverage as you go from 1x36 to 2x76 to 2x101 to 2x151. And basically this data coupled together with the 63-degree data that I showed on the previous slide is where we now recommend that if you are doing surveillance in the RUO mode, so once again, this is not about detection, these are all PCR positive samples that we know the virus is in there, your best chance to get a really nice complete genome is to run this with at least 1 million reads, as I'll show in a second, and having coverage of 2x100 or more. This tech note that we put out recently describes that in a lot more detail, and it includes this this heat map here that shows the kind of the 1x36 data here is in the first column all the way out to 2x151, and you can kind of see the degree of genome coverage. As I said before, the genome coverage at 1x36, even at 1 million reads, is not that bad. It's almost 98%. But in order to get it up to 99, and really essentially 100% coverage, it doesn't hurt to do longer reads. And so that's what we're encouraging everyone to do, the 2x100 at the minimum. At the same time, we've been working on these surveillance optimized biochemistry. We also have been optimizing our informatics tools that we recommend for this. So in this chart, it shows some of the data analysis tools that we have created specifically to analyze COVID and COVID-seq data. And the top row there is what we use for detection. That's the EUA approved COVID-seq software. It runs on local or cloud option, and it is used exclusively with the COVID-seq test kit. In fact, it's specified as part of the emergency use authorization that you have to use this software together with this kit. And of course, it's very prescriptive as a diagnostic. In the surveillance mode, the RUO mode, we can be far less prescriptive and allow people to change the protocols as we've already talked about, the read lengths, and also the analysis methods that are used. And so that's where we've developed COVID-seq RUO, and also an app that I want to spend quite a lot of time talking about, a relatively new one called COVID Lineage app. This one is really quite interesting because this is really kind of a true surveillance tool compared to just a detection tool. And the other thing I want to point out is Illumina has made a commitment to make all of the COVID-seq tools free of charge. So the COVID-seq EUA tool, the original COVID-seq software, is run on our Dragon server and also in our base cloud computing environment. So it can be run either locally or in the cloud. It runs very fast, and it does extremely fast demultiplexing. This is quite a lot of samples on a single run of a NovaSeq, and that demultiplexing alone, believe it or not, can take quite a lot of time. But Dragon does a really great job of speeding that up. Then quality checking of all the samples, then viral detection, and then mutational identification and genome reporting, like I said, was sort of not a critical part of the EUA. it was kind of treated as bonus information at that time. This is the app itself. It's different versions. You can, like I say, you can run it on Dragon or in base space. And this is the output of the report that we were delivering from the EUA test. It has a lot of metrics based upon the controls of and the logic of the controls. This is a human sample, so it should have positive human PCR results. But then if the positive signal comes from the human signal, then it will go and look for the virus, and does the virus signal reach that threshold of detectability that I was talking about a few slides ago. So does it pass all the QC? Do we detect more than 90 amplicons? If so, a sequence was reported out. And then based upon a sufficient number of amplicons, do we detect enough to call it detected? And essentially that was 5 out of the 98 amplicons showing up, we would call it a detection. The COVID Lineage app, like I said, is a relatively new one. This is a much more flexible app than our original COVID-seq app, which I said, like I mentioned, was designed to be part of a diagnostic test, and so it's very stringent in the way it could be used. This one is much less stringent. In fact, you can analyze data from any instrument at any depth, any read length, and frankly, any library prep. This works with RNAseq data, enrichment data, the COVID-seq test, or homebrew Arctic protocols, or any other Amplicon protocol that's out there, or any other RNAseq protocol that's out there. As long as you have FASTA files, so you run our sequencers and you have to run at least BCL to FASTQ files. So as long as you have FASTQ files, you can enter them into this app and then do these types of analysis that I'm going to talk about. As of today, we only have a cloud version of this app available. We are going to be releasing a Dragon version of the COVID Lineage app here in the next few weeks. So this app, like I said, is much more flexible. It allows you to actually tinker with the thresholds for the detection thresholds and also for the sequencing metrics. So you can adjust coverage thresholds, virus detection threshold, virus negative threshold, human control threshold. You can change those any way you want if you're using it for detection in an RUO sense. It also allows you to basically create FASTA files from the sequences that can then be used in all kinds of interesting analysis. So as I said, if you look on the left here, you can take the output of the COVID-seq app or just FASTQ files from any Illumina sequencing run that has gone from BCL to FASTQ conversion, or from other applications that we have that I didn't talk about today, but like, for instance, our respiratory virus oligo panel, which is our kind of a pan respiratory virus, has up to 30 or 40 different viruses that can be detected there. If you have data from that and there are SARS reads in there, you can take those FASTQ files and put them through this app as well. And so this app creates a FASTA file, which is the genome sequence, and then that kind of under the hood runs a series of other applications for you that are open source tools that we can kind of utilize, that many people around the world are using these tools right now to kind of characterize the virus during surveillance. Also, these FASTA files can be used, if you look at the lower arrow there, for sharing, data sharing, like submission to GISAID or SRA submission or SRA import. So I'm going to talk a little bit about some of the details of these. So this is the Lineage app summary report, and it's a little bit different than our original COVID-seq test app summary report. In this, we, like I said, most of the time the samples that go through this are known to be positive. So it's more of a question of what is the lineage and what kind of variants are found here. So one of the handy things that this does is it can take all of your samples, and instead of having to load them one at a time into the Lineage report, you can upload them as a concatenated FASTA file, which is quite useful. It also has a link to Nextstrain where you can upload it directly to Nextstrain. It shows you the lineages that are assigned by this pangolin tool. It gives you probabilities of that. The version of the pangolin tool is updated at least weekly these days with new data and sometimes new lineages. It gives you some QC information of that, and it kind of tells you the percent of genome that is covered that went into that analysis. Here's an example, for instance, just to prove, oh, I want to go back one slide. One thing I wanted to highlight here, if you look at the 5 data types that we're putting in here, none of these are actually the the classic COVID-seq data. These are all examples of other types of assays that have created data, FASTQ files, that have been loaded into this COVID lineage app and analyzed this way. So, like I said, this app can analyze data from any application, any library prep, and any read length, and give you out this kind of information. Here's an example, for instance. This is an RNASeq, so this is like shotgun metatranscriptome of a nasal swab. And you can see here's the FASTA file that was created by the COVID lineage app here on the right. And it will also show you a coverage plot of how well the 29,000 plus genome of the virus is covered. And you can see we have excellent, in this particular example, 5,400 average median coverage or median coverage. So this is just an example of one of the outputs. The other thing it will do is report out every single Amplicon that was used, and out of the Arctic in this case, or our human controls. You can see this was a prep that didn't use since it was an RNA-seq type prep, did not have our human control Amplicons in there, and so there was not very good coverage there, but we do kind of report out what the coverage would look like in the Amplicon sense across these various regions as well. So that's interesting. And you can use this to kind of compare across different preps or across different types of Amplicon assays, et cetera. And like I said, under the hood, the Lineage app will take your data and run the pangolin algorithm on it, and then report out this sort of information here, where you can either look at it on the pangolin website, or you can look at the summaries that I've showed you before here. This is just showing in the pangolin website what it looks like in terms of regional distributions, phylogenies, and this is this sort of information you get for every single positive that you put through this application. And finally, the other really useful new tool is called Nexclade. These are from, this is a takeoff from the group that created Nextstrain, but in Nexclade, they get into really a lot of detail about the mutations, once again, the lineages that they would assign, the mutations that are found in each genome, which kind of amino acid substitutions those are, and amino acid deletions that would represent. And you can also download these to Excel files for further analysis, or you can compare the phylogenies like in the Nextstrain type sort of trees that we've all gotten used to looking at over the last year. So I'll just close with that. I think hopefully I've convinced you that studying variants and understanding how this virus evolved is going to be really, really important for the next phase of this pandemic, and what our response is going to be to vaccinations and unfortunately lack of vaccinations in other parts of the world. And we have to kind of keep our eyes out for what kind of variants are evolving out there at all times. If we don't understand the variants, I think that the less we understand about them, the longer this is going to take for us to get through this pandemic. So hopefully this has helped you understand how next generation sequencing is being used to characterize and understand variants. And with that, I would like to thank all of you for your time and attention, and hope you enjoy the rest of the conference.